Science Rendezvous Hackathon. We're just going to get started now. So yeah, give me one moment. Okay, so welcome everyone to our Innovation Speaking Series number 1. So this is our first event with Science Rendezvous Hackathon. And we'll talk more about what other kind of events and learning activities there are. But I'm really happy that you guys are all joining us today. So a little bit about who we are. So I'm Douglas, and I'm part of the Science Rendezvous executive team, and I'm leading this kind of hackathon for this year. And it's a really cool idea because we've never done this before, and it's of course all virtual. So it's been quite an adventure, and it all started from a whiteboard where we just had the idea of a hackathon. So that's that. And then we also have Eduardo, who is leading the computer science or programming kind of competition. So Eduardo, you want to say a few words? Oh. Okay, I think he's going to join us in a bit. No worries. So our schedule right now is we're going to introduce our SR Hacks and what kind of events we're doing. I'll do a quick activity with you. And then at 10:45, we're going to have our first speaker, Dr. Michael Reber from the Kremlin Institute in University of Toronto. And then we have a 10-minute break. And sorry, this is a typo, but we have Dr. Francis Skinner, who would be presenting second. And then we have our career panel discussion at the end. So if you have any questions throughout this talk, feel free to type on the Zoom chat, on the Q&A, or if you're a student with us, go in the Discord and we will have people monitoring there as well. If you have any technical issues, please feel free to email us at uoftsr.virtualhelpdesk@gmail.com. So again, make sure you type in the chat or the Q&A and someone will be able to help you there. So a bit about our team. We are made out of undergraduate students or graduate students doing their master's or PhD at the University of Toronto. And what Science Rendezvous is, it's been 14 years of existence, I guess. And our goal has been creating a large-scale science festival, one of the largest around the world, where we engage students and the public on different science activities. So we used to have a bunch of departments at St. George Street doing all these activities, from rockets and fruit launchers and things like that. So this year we had to do it virtually because of the pandemic, but we still had over 1,500 attendees, which is amazing, within our U of T festival. Of course, there's others out there. And this year we were doing something completely new that we were thought out by the Science Rendezvous outreach team, because we were trying to engage the public outside of the actual festival day. So what we have is a 3-week hackathon program for students grades 6 to 8 within the Greater Toronto Area, where they will be competing to create an innovation either through programming or through engineering, and then submit it to our esteemed judges. And the top 5 finalists from each competition will attend will speak at our SR Hack-Off Day. So this is the big closing event of our program, and we will have them talk about their innovations and ideas to the world. We also have a bunch of learning events that will help students understand basic concepts of engineering, as well as computer science. So anyone of any experience can join in our events. So if you haven't already, you can still register with us until June 20th. So go ahead to our website at www.sciencerendezvousuoft.ca/srhacks. So for our learning events, we have 2 different types. So today is our Innovation Speaking Series. We also have another one on June 27th. And the idea here is to bring real scientists, engineers, or entrepreneurs who have created startups and things like that, who are researching or innovating or creating new things within the computer science field or the engineering side of things to change the world. And today we'll be talking with Dr. Michael Reber and Dr. Francis Skinner, who are using computer science to really change the way we're looking at human diseases or human biology and so forth. And then on our 27th, this is our second innovation speaking series. The time is still to be confirmed, but we have Sheila Bagiana from Free O2 Foundation and Shishi International, who is a biomedical engineer creating technology to develop oxygen for hospitals in need in Uganda. And we also have a mystery speaker, which is to be announced, but hopefully it's not totally confirmed yet, but we'll get someone from the automatic or AI and car industry. So that would be really, really cool. Okay, and in terms of our workshops, so again, our program is designed for anyone of any experience, and not just students, but anyone in the public can attend our innovation speaking series as well as our workshop. So we have our first one on June 12th, which is a Microsoft MakeCode, and it's a bit of block coding and basically teach you how to do beginner programming and programming a microbit, which is this cool kind of hard drive chip. And then we have our engineering design, which will talk to you about the basic concepts of engineering. And finally, I will be leading a hydraulics machine workshop where we'll be creating kind of hydraulic machines in a DIY kind of workshop thing. So again, anyone can attend. And make sure you guys also attend our Hack Up Day, which will showcase a bunch of different student projects and our top 5 finalists from our event. So this calendar or schedule is more for our students, but in blue are all the workshops that you guys can attend. Reminder to students, we have our opening ceremony and information session today at 1 to 2 p.m. So right after this amazing event, we'll talk to you about all the details, sorry, the details about the competition. So please make sure to join us there. That one's only for students and not for the public. Okay, so before we begin with our amazing line of speakers, I just wanted to do a quick activity with everyone. So this is something that I remember talking about as a kid with friends. So imagine if you were stuck in an island and you were the only one there. What would be one item you would take to the island? So feel free to type in the chat and I'll give you a couple minutes for this. And I promise you there's a reasoning and there's somewhere that I'm trying to get with this. So feel free to put in. There's quite interesting answers. Sisters or family. There's a flare gun or a hot pot. So these are tools that you need maybe to survive. A water purifier, knife toolkits, etc., etc. A charged phone. Yeah, that'll be good for, I don't know, 8 hours until your phone dies. But an accelerator, internet signal and radio. So that would be really cool. Okay, awesome. So keep typing in there. These are really great answers. Oh, someone said a book called How to Survive on an Island by Yourself. That's amazing. So here are some of the things that I thought of. And I remember back as a kid, I've said those things too. So I would bring, for example, an airplane where we can just get off the island. And I know that might be cheap and cheating a little bit, but that's my answer. And airplanes were really developed by the Wright brothers way back then in the 19th century. And they just pieced together these wooden planks and an engine and just started flying. That was the beginning of aviation. And then till now, we've developed airlines and crop dusters, that help us with farming and agriculture. You can travel across the world anywhere you want to in less than 20 hours. And the aviation industry has just exploded, and it's amazing. Um, and another thing that I would have maybe bought was an irrigation system. So for people who don't know, these are water pipes, and this was developed way back in medieval times or even earlier, where people use piping systems to control the water in the farms so that you can create large farms and food and so forth. So this really helped economize and globalize our market and connect people around the world, but also feed and provide for a city. Another thing maybe I would bring is 3D printers that use batteries. Of course, I can build anything I want. And this was developed in the 1980s and has really exploded in the uses that we could do. So you might ask, okay, Douglas, like, why are we talking about this? Like, who cares what you want to bring in an island? Um, I think all the things that you guys have mentioned, they're innovations, they're tools, there's things that someone somewhere has developed. Maybe it's a scientist, maybe it's an engineer, maybe it's a random guy or lady in a house just having an idea and made something themselves. So innovation really comes anywhere and starts today. It starts with you guys right now. And these are innovations of the past. And it's developed incredibly to have kind of toolkits of the future. So there are a bunch of companies that are starting to do drone delivery systems where they go around to rural areas to provide medical aid and tools and food and things like that. So I remember Google X Moonshot developed one of these companies, and now it's expanding. We also have systems where we can actually plant in the desert. So I think Desert Control is a startup who made soil where people can actually fertilize the desert soil. So now it's growable for crops, and that can help us fight world hunger. Or we have 3D printers that have evolved to print entire houses, and this can help our rural communities around the world and fight off homelessness and things like that. So really, their innovations are constantly growing, and it's up to us, it's up to you guys to really think about what kind of problems and challenges you want to fight off or tackle, and use kind of innovation, including computer science and engineering, to do these things. Now, I'm almost done, a couple more slides, but it's very complex too. So when you think about just a drone, think about in the chat, type in all the things you think you need to run a drone. So it's a lot more than you think. On the engineering side, there's electrical systems, there's propulsion and flight systems, there's what materials you use on the drones and how you can make the most aerodynamic. There's sensors, someone said, so that can help with collision avoidance. And maybe you have programmers who can make these automatic flying systems. And so these drones can detect where obstacles are and kind of guide itself. And then you have other fields that it collaborates with. So meteorology, or study of the weather. You have geology, where people are using drones to map out archaeology sites or different volcano sites to see what's happening. They're also using it to use these thermal cameras to see which plants in a farm are healthy and which plants you have to fertilize more. So there's a lot of collaboration across different fields. And of course, there's the business stuff who I have no idea what's going on over there. It's a little black box in my mind. But all the management of a company, of looking at the financial systems and human resources and marketing and so forth. So all of this all comes together to develop a product that a company can create. So it's very interdisciplinary. And it starts with you guys. It starts with our new scientists, engineers. So scientists are people who understand how the world works. And we have Dr. Reber and Dr. Skinner, who are scientists, and they'll talk more about their fields. We also have engineers who build the tools that we need and create things. We have our programmers who do a lot of coding into the computer systems and data analyses. And we also have our entrepreneurs or our business people who pitches the models, create ideas, and so forth. So there's, even within these different categories of careers, there's a million others within a scientist. There's heart scientists, there's brain scientists, there's biologists and plant biologists, all these different areas. So I really encourage you guys to explore all the different types of careers, and also think about what problems you want to solve. Is it hunger? Is it poverty? Is it sustainability and global warming, or a pandemic and kind of health and disease? Or maybe it's to create systems of clean water, filtration and things like that. And it really starts here with you guys. So I would encourage everyone to um, attend TED Talks, attend events like this, learn new skills with coding, start researching, reading a book, picking up something that you really like, really going for it, and talking to people in the field. So we have, again, amazing people in the Innovation Speaking Series where you can talk to, and hopefully we can connect with. So I ran a little bit over time, but I'll leave it at that. And so I'll give it to Eduardo and he can introduce our first speaker.
Yep, thank you so much, Douglas. And this is what we're doing the speaker series, is to kind of help everybody to try and understand how skills such as computer science or programming can feed into leading to developing new technologies to help people. And one of the persons that are going to teach you a little bit more about that is actually a neuroscientist who received his PhD in human genetics at the University of Diderot in France, then continue on to finish his postdoctoral work at the Salk Institute in San Diego, then established his research in the development of the visual system as a professor at the Institute of Health and Eye Disease in Strasbourg, France. He now continues his journey at the Krembil Institute here in Toronto, and one of his projects revolves around using virtual reality as a new therapeutic method to help patients with visual deficits see better. So here to tell us more about that research, please help me welcome Dr. Michael Reber.
Hello, Eduardo. Hello, everyone. Can you hear me okay?
Yep, sounds good on my end.
Okay, so should I start to share my screen?
Yep, you can share your screen. Awesome, it looks good. I think you're muted, Dr. Reber.
Yeah, I'm just, there we go.
I'm sorry.
There we go.
Yep, that's good.
Is all okay?
Yep.
Okay, good morning, everyone. I'm very happy to be here. I want to thank Eduardo for this nice introduction. And I also want to thank Science Rendezvous for inviting me to talk to you about the work we are doing in the lab. And I also want to thank you all for attending, because taking some time on a Sunday morning, a beautiful Sunday morning, that's really nice of you, and it shows that you're very much interested in innovation. So I'm going to talk to you a little bit about one of the research projects we have in the lab, which is trying to find a way to help people with vision problem using a very unique and innovative approach, relying on virtual reality and the Oculus Quest. So what does it mean having vision problems? So here is a diagram of the primary visual system in humans. So visual information is captured. Can you see my pointer here?
Yep.
Yep, okay. So visual information is actually captured by the eye, and then this visual information is carried over to the brain, to that blue area here, which lies in the back of your head. It's called the visual cortex. And visual information is carried over through those, some sort of cables that we call optic nerves, and that are basically carrying the information to that area. So all along that pathway, you can have bad things happening. For example, in the eye, and in particular, the retina, which is a part of the eye containing cells that are capturing the visual information. In that area, in that organ already, you can have bad things happening. And you may have heard of some of those eye diseases that are actually affecting the retina, such as glaucoma or age-related macular degeneration, also called AMD, or diabetic retinopathy, but also trauma. You know, if you get hit in the eye by a ball or a puck or something like this will damage your eye, and eventually you will lose vision. Another area where you can have damages is the optic nerve. So the optic nerve, imagine you have a bundle of electrical wires, and these wires are connecting the eye to the brain. So all the visual messages are going through those wires. So imagine that you are somehow pinching these wires or cutting halfway through this bundle of wires. The visual information will be impaired, you know, so part of the visual information will be missing, or eventually you'll have, you're slowing down the message and the way it's transferred to the brain. And this can happen after trauma or cancer, brain cancer or glaucoma. And then the third area in the brain where bad things can happen is the brain itself, actually, and this visual area in the back of your brain. This area receives visual information and actually performs the decoding, the analysis, and the processing. And so trauma, cancer, and stroke can affect this process, and therefore impair somehow the analysis of the visual information. Knowing that this area is crucial, because once the visual information has been decoded and analyzed, it's sent throughout the brain to other areas, such, for example, your memory. Because if you enter a new place, you know, you want to know whether you've been there already, so it's familiar to you, or it's a totally new place. So this image is then compared to what you have in your memory. So as you can see, I mean, there are many ways to somehow create vision problems. There are many ways to disrupt the visual information, whether it's a trauma, cancer, stroke, or an eye disease such as glaucoma or AMD. But what does it mean having vision problems? What does it mean having a troubled vision? You know, besides blurry vision that can be corrected with glasses or contact lenses, or when you have your lenses in the eye, which becomes opaque, then you have cataract and you have surgery for this that can fix this. But glaucoma, AMD, diabetic retinopathy, those are diseases right now we cannot cure them. You know? So we can slow down the disease, but we cannot cure them. So what does it mean when you have glaucoma? Actually, what do people with glaucoma see? Or what do people with AMD see? So if you have normal vision and you recognize this video right away, you know, it's familiar, very familiar to many of you probably, Minecraft. So if you have normal vision and you play Minecraft, this is what you would see, okay? Now, after a stroke or a cancer or a trauma, well, you may experience something like this, which is called homonymous hemianopia. And you can have your left field or your left visual field missing, but this can also target your right visual field, or this can also target part of your visual field, okay? But you can easily imagine that if you experience something like this is life-changing. You know, whether you want to go around busy places or bike or drive, this is now nearly impossible. And something like this can happen very quickly, very suddenly. You know, a trauma at the back of your head can generate visual problems like these ones. Patients who have glaucoma, for example, they have a shrinking of their visual field. You know, they are not able to see the size of their visual field anymore. And this is a dramatic issue also, you know, because you can imagine you can't drive or bike around anymore because you are not seeing what's coming, the incoming traffic. You know, you won't be able to see it. What are people with macular degeneration, you know, AMD experience? Well, they have a loss of vision in their central visual field. So they don't see the center of their visual field. And this is a major issue for reading, for example, or recognizing faces. So as you can see, I mean, these visual problems are definitely life-changing, I would say. This is a major society, health society issues. I mean, if we ask Canadians what is the most feared disability, 70% of them will answer vision loss. So this is a major issue. So what can we do? What can we do to help people who have such visual impairments and so-called low vision patients? Well, I told you earlier that the brain receives a lot of visual information, and actually 50% of your brain will at one point deal with visual information. Now, if we draw a diagram of the visual system in the brain, this is what we would get. So down below is the eye, that's corresponding to the eye, but all the rest is brain. And so all those little squares are different brain areas which are dealing with visual information. And all those black cables and red cables are actually neurons and axons that are connecting those areas together. So this is a very complex and organized network, built in our brain to connect all the brain areas that are dealing with visual information. So after a stroke or a trauma or a disease, you may affect part of this network, right? So part of this network might be impaired. What really happens actually is that in this affected area, you still have some cables that are maybe lying there, but they are not connected anymore. Or you may have brain cells in that area which are also disconnected, but they are still alive. If you have like larger areas affected, you may have within those areas cables lying disconnected, but they are lying there, and you may want to try to reconnect those to the major grid, to the major network, right? Or well, you may also want to create a bypass somehow, to reach this area over there, 7B. So you would have to create some sort of shortcut that goes to 7B, avoiding that affected area. So this can all be possible. This reorganization of the brain is actually happening every day for you. As soon as you learn new things, your brain is reorganizing and rewiring. Whether you are learning a new lesson, a new skill, a new technical movement because you play sports or whatsoever, your brain is constantly adapting, rewiring. And this process is called neuroplasticity. And you may have heard of this word, maybe. And this happens all lifelong. So for sure, it's faster and more efficient when you're young, but adults and even elderly people also have the ability to rewire their brain. It might take a little bit more time, but it's still possible. So we really have to consider our brain like a muscle, So we should find ways to basically train the brain to reorganize this wiring in certain ways. But if you want to train your brain, as often, you need a coach, right? So basically, we are the coach, and the tool we use is the Oculus Quest, sorry. So now if you would wear the Oculus Quest, and this is what you would see. So this is one of the visual tasks we are actually developing in the lab for those patients. So here in the middle, you have a green spot on which you are supposed to focus your gaze. Okay, so basically you are looking at that spot. And then you have 8 yellow balls around here, 8 yellow spheres. So what you will see on that video is that one of that sphere randomly will turn red, and then this sphere will turn yellow again. And everything is going to move for a few seconds, and you are supposed to keep track of the sphere that was red. So we call that the cued target. So you're supposed to take to keep track of the cued target. And after a while, all the movement stops, and using the laser pointer here, you will select what you think was the cued target. Okay, so this seems very easy for you probably, and it is actually if you have normal vision. But just imagine if you have hemaniopia, or just imagine if you have glaucoma. This suddenly becomes very challenging. So what we have done is that we generated a sound so that the cued target actually emits a sound, which allows you to track this target using both your eyes, but also your ears. And this is basically what's happening every day. Every day when you go out, you use your eyes and your ears to detect what's going on around you, to detect any incoming bicycle or car or so on. So you're using those two senses together to basically apprehend your environment. And we are recreating this natural behavior in these training, some sort of perceptual learning, brain training, to force the visual system to rewire certain ways. We can make it even more realistic or even more challenging for people with low vision. Here is an example of the exact same visual task, but not on a black background, but a more realistic background. So that's a video we recorded at the corner of Dundas and Spadina here in Toronto. And so we are adding this as a background. So basically we are putting the patient here in some real life situation, and they keep training with those goals for their visual stimulation procedures. And so you can imagine if you have hemianopia or glaucoma, this is what you would see. But this type of training comes after the first step that I just showed you with the black background. So we first train the patient on the black background, see whether there are progresses are made, and then we move on to more challenging and more complicated features, such as this one. But of course, to generate this, well, we need programming, right? And you are probably all familiar with this, the Unity platform that we use to create those games. And so here I show you a short video of the Unity editor that we use and some of the features we use to create the ball, to create this virtual space, to generate the sound, bounce the sound to the ball, and so on and so on and so on. And so we use this to create those visual tasks. And then we test them to make sure there are no bugs, and so on and so on and so on, and to fix little things that may go wrong. And so, but you can imagine that. So we generate this, and then we put it into the Oculus Quest. Now, I was saying this is some sort of a training, right? So if you train, you have to do this in a regular pattern, right? So you have to do this every day or every other day or so for some long period of time. And as you can imagine, patients with low vision will have a hard time coming to the hospital or coming to the lab and coming in to train for like an hour or so every day. And this would be very challenging for them. So what we do is we provide these Oculus to the patients. So basically the patient takes the Oculus and go home. And we basically, from the lab, we control the Oculus and we control the program. So here is the remote dashboard, Unity remote dashboard. And this allows us to control every single parameter in the Oculus, whether the number of balls, the number of cued targets, the speed, the duration of the exercise or the stimulation. All these can be controlled directly from the lab. And this is then sent out through Wi-Fi to the patient's Oculus Quest. And the patient can nicely train from home without having to leave and to go to the hospital. And this typically includes coding, right? So that's the purpose of this session today. And here is just an example. So we use C# as a language, and here is just an example of the coding that was made. And this is actually part of the coding that is binding the sound that I mentioned earlier to the cued target. So we are here in this code, we are binding the sound emitted by the cued target to the actual target, so that when it moves, you can still follow it in space. So how does this all work? So basically we are in the lab and we are coding, generating those visual tasks that are then sent through Wi-Fi to the Oculus Quest at the patient's home. And then the patient follows the training program. So right now, the training programs are typically 15 minutes to 20 minutes every other day for 6 weeks. And each time the patient is training, we receive real-time data from the Oculus Quest in the lab. So we receive a lot of data about the performance of the patient. So how many times the patient was able to detect the target, how long did it take, what was the duration of the exercise, and so on and so on and so on. So we receive a lot of data real time that we process and we analyze, and we then modify the code accordingly. So we try to modify the code so that the overall exercise of the overall stimulation is following the progress of the patient. So that for the patient, it's still challenging each time the patient is trained, right? And this somehow maximizes the efficiency of the training. And so basically, how would that work? You have vision problems, and so you would go to your ophthalmologist or see our colleagues here at Toronto Western Hospital. And if they believe it would be relevant for you to follow one of these visual stimulation programs, they would include you in our research study. And then you would perform this visual stimulation for several weeks at home. And after the training would be done, you would go back to your ophthalmologist and redo the same tests and hope that your vision improves. And actually, we have preliminary data about to be published very soon on a few patients with anonychia, and we see that this type 6 weeks training like this visual stimulation improved dramatically their visual perception. Okay, so I'm reaching the end of my talk. I want to thank the people involved in this work, and one of them is one of our hosts today. So the work that I presented here is 98% done by Eduardo. He's the one who did all the programming and all the creation of that visual stimulation in the Oculus Quest. We are very happy because this work was showcased in a recent special edition of the Globe and Mail with the Vision program at the Krembil, and our work was showcased there. That was in September 2020, I think. And for the summer, we have a student joining the team working on that project. So the student is Pai Nazir. It's a third year. He's a third year computational science student at the University of Toronto, and he's joining us to help Eduardo further with the coding and improving and modifying all those visual tests we're performing. All right, that's it for today, and I'll be happy to answer your questions. Thank you.
Thank you so much, Michael. We're actually going to hold on those questions until the Q&A. And for the people that have asked questions, we will refer back to them once at 12 later on in this presentation.
Okay.
And yeah, so thank you so much again, Michael. That was a fantastic presentation. So I think now we're going to move on to our 10-minute break. Is that correct?
Yeah, so we're a little early, but we can start our break now. So be back by 11:20, and we'll go from there. Awesome.
All right, so we're gonna get started and introduce our second speaker, who is actually a computational neuroscientist, whose passion for mathematics led her to pursue her PhD in biomedical and mechanical engineering at the University of Toronto. From there, she moved towards completing her postdoctoral work at the University of Brandeis and the University of California in the United States. She now continues her research as an independent scientist here in Toronto at the Krembil Institute, where she focuses on understanding the biology behind how the network of brain cells work through the programming of predictive mathematical models that seek to answer very complex questions about how the brain functions. So here to tell us more about her research, please welcome Dr. Frances Skinner.
Okay, can you hear me?
Yep, sounds good.
Okay, unfortunately my background has disappeared, so hopefully
Oh, no, that's
it
okay.
worked there. Okay, all right. Okay, so what I'll do is I'll just put on a slightly different background for now. Yep, okay. Okay, so let me share my screen. Okay, you have to stop sharing your screen, I guess. Yep, okay. Okay, and okay, and can you see that? Screen share?
Yeah, looks good.
Okay, so, yep. Okay, so should I go ahead?
Yep, floor is yours.
Okay, okay, so thank you very much, Eduardo, for the introduction, and thank you very much for the invitation to speak here today at Science Rendezvous Hacks. To all the organizers, having organized things myself, I know there's a lot of work that goes on in the background, so thank you for that. And the title of my talk, as you see here, is It's All About the Youth. And since I don't typically talk to this audience, but I thought this was a appropriate title for a couple of reasons. It's in quotes because it is something that my Uncle Sally said many years ago. Him and my father were sort of struggling to open a can. I think it was a bottle of jam or something, and they couldn't get it open. And they called my son over, who was probably about 15, so the age of a lot of you students, and he just went boop and opened it up. And my Uncle Sally said, "Oh, it's all about the youth." And they laughed about it. I keep laughing about it nowadays. So that's why it's in quotes, and I think it's very appropriate for the audience today. So I stole it. So okay, so I'm Frances, and here you see a logo, Skinner Lab, because we studied neurons and microcircuits. But I wanted to mention this part here, which is about developing bridges, which is really how I see the work that goes on in our lab. And so what I'm going to do in the next several minutes is tell you a little bit about me. Let me just advance my slide here. Yep. So a bit about me. So here I am in cartoon form was created by one of the admins here at the Krembil Research Institute. And this is where I typically work at the Krembil Discovery Tower, which is part of attached to the Toronto Western Hospital in the corner of Bathurst and Dundas. And this is me in one of my favorite places by a waterfall. And this happened to be in Iceland. Now, this is where I typically work, but these days, this is where I work at home, because as you know, we're all facing these challenging times, but I'm very fortunate because of the computational mathematical work, so I can actually carry out a lot of my work at home. But just to tell you that I hail from Trinidad and Tobago, and this is one of my favorite pictures because this is what you see when you get off the plane in Tobago and walk down and you see Store Bay Beach. So that's where I'm from originally, where I grew up. And of course, this is me in 2019, and I don't need to say anything more. Okay, moving on. Okay, so a bit about what I do and how is what I thought I would share today with you. So I'm a neuroscientist, and for those of you who could see my picture, wait, there's the camera. What I'm doing, I'm holding a little brain in my hand, a squishy brain. And if I was giving this talk physically, I would probably throw squishy brains at the audience, but I can't do that. So I'm just gonna sort of throw it up in the air. So I'm a neuroscientist, which means one of the many people who want to understand how the brain works. And I put this set of equations here. These are the Hodgkin-Huxley equations. I put them here because, first of all, I'm a neuroscientist, but I'm actually a computational neuroscientist, so mathematical neuroscientist person, which means that I use math and computers to help us understand the brain. And this is an image that is very Toronto-centric that I wanted to share, because what you see is what used to be called a Sky Dome has a brain. And it's just a silly joke. And this is the CN Tower that is sort of like an electrode that you might inject into a brain cell for experiments that are done. This image was actually made by my husband, and this is actually part of the early universe. But I think it makes a point that, it's what the point I want to make is that really neuroscience is a giant collaboration. And so this image was made for a conference that I organized many years ago in computational neuroscience. Okay, so being a neuroscientist, we want to understand how the brain works. And what we mean by that, and you heard the previous speaker talk about codes in the brain, so our visual system works. And of course, we have many other systems and brain structures. And so how is it that the brain does what it does, allows us to move, hear, think, have memories and all the rest of it? Well, it seems clear nowadays that it has to do something with oscillations, that is rhythmic activities in our brain. And that's because many years ago, actually back in 1929, Hans Berger actually recorded from the surface of the brain electrical activity that was oscillatory, so rhythmic. And this is a book, and there's been many books, but this one I point out called Rhythms of the Brain, that was written by Yuri Bizaki in 2016, which talks about all these rhythms, different frequencies that record in the brain. And importantly, Bob Marley, whose music I grew up listening to, said in the 70s, we got rhythm. So for me, that nails it. The rhythmic activity is what brain codes are all about. So once we talk about brain oscillations that we know are there and we know are important, we want to understand how they're generated. And how they're generated is because all these different cells and networks that exist in our brains. And in particular, here's an image of one particular cell type in our brain that we've actually modeled. And I'll maybe mention it a little bit more later. But this is sort of to bring home at this stage that we have many different cells and many different networks, but they're highly complex. The brain is the most complex organ in our bodies. And so this is a really challenge that we have to face if we want to understand the brain workings. And this is why it's so important that math and computer science sort of come to the fore. It's a giant collaboration to really help us figure out what's going on. Okay, so here's the human brain in this case, and we can record electrical activities in this example here. So this is sticking an electrode on the surface or inside the brain, depending the type of recording you're doing. And you see this rhythmic, so this repeating sequence of events, electrical activity. And the part of the brain that we focus in our lab is called the hippocampus. So this is a structure deep inside what's called the medial temporal lobe. And it's highlighted here in blue. And why it's studied by many people is because it's actually associated with memory, learning, has rhythmic activity, and importantly, you can do experiments on it. So on the left here, you see a coronal slice of the human brain, and this yellow part is the hippocampus. If we now take a sort of a banana slice of the hippocampus, this is what you would see. And this is just to point out the beautiful structure, the laminar structure, all the different cell types and circuits that are formed. This is a rodent or rat or mouse brain on the right here, and this is the hippocampus. If you take a similar slice, you see a similar structure in terms of different regions of, they're called CA1, CA3, DG in the rodent as in the human. There are differences in the details, but you have the same range of different cell types and circuit structures. And so this I'm showing you below here is what you would see if you actually stuck an electrode in the rat brain down through the banana slice, and you see this ongoing electrical rhythmic activity. And you see that it's not just a smooth sinusoidal, it has sort of structure associated with it. And depending on exactly where the location of the electrode is, it varies. So I just mentioned this to sort of bring about sort of the richness and the dynamics of sort of these recordings in the hippocampus, as well, of course, other brain structures, visual cortex, prefrontal cortex, all the rest of it. So these rhythmic activities that I showed you, there's one type that's called the theta rhythm, which is the strongest rhythm you would see if you record in the hippocampus. And this rhythm, as I'm sure, as is schematized here, is of a frequency between 3 and 12 hertz. And why I'm showing you this rhythm, and it's pretty famous and well known in the field, is because we now know, and actually a Nobel Prize has been associated with it, that this rhythm and the sequences of firing of individual cells signify a code, so a phase code of how the animal navigates in space and actually associated with memory too. So what you're being shown here, because of these experiments, of course, can be done in rodents and not directly in humans. Although I must say recently, thanks to virtual technology, it has been shown that this similar kind of code does exist in the human brain. But basically what happens is as the animal moves through, some location, you have a firing, and these are these different colors, yellow, green, blue, signifying where the animal is physically. So the code is individual firing of cells relative to this ongoing population activity. So basically you have this frequency of 3 to 12 hertz, and individual cells in the hippocampus, and they're called place cells, fire at specific times during this cycle to create a code that people refer to as phase coding. So in other words, this rhythm is not just some rhythm we record, it actually has meaning in terms of memory, spatial navigation in the animal's brain. So it's pretty exciting to think that this code, this rhythm can give us understanding of memory. Now, the challenge is we know it exists. We also know that it changes in behavioral, pathological states, but to understand how it comes about in the most complex organ of the brain is sort of what we sort of dedicated a lot of effort to. And the reason it's challenging is because, as I mentioned, you have lots of cells and networks in the brain. You have actually as many cells in your brain as there are stars in the Milky Way. And these cells are of different types. So here you see a schematic of 6 different cell types that are present in the brain. And they're distinct in terms of what they look like, morphology, how they're connected to each other, what they stand for. And if you stuck an electrode in this particular cell, it would have a distinct firing property. So they differ in these ways. And somehow they come together to create this population activity, this theta rhythm here that I referred to, that's important for spatial navigation, memory consolidation, and the like. And if we look at, say, this particular cell type, which happens to be the one that I showed you in the beginning, that has that particular structure, it's called an OLM cell, Orion lacunomolecular cell type in the hippocampus, you see it fires distinctly at this time during the ongoing theta. So sort of in the lower half as it's coming down. And you see other cell types doing something. So here we have these individual cell types doing something particular during this population rhythm that is meaningful for the animal's code. So what we try to do in our lab is model this to try and get an understanding of what's going on. But what's hugely challenging that I've sort of tried to sort of bring about is that our brains are very complex. It's a multi-scale mental temporal organism. You cannot do experiments simultaneously at the behavioral level, at the macrocircuit whole brain level, at individual local circuits, at individual cells, at connections, at the molecular genetic level. Experiments occur, but not at the same time. So this is where mathematical modeling and computational neuroscience, and of course, when I say computational mathematical modeling, by definition, that includes coding and programming. I'll come back to that in the next slide. But what I wanted to show you in this slide was just so that all the different levels that we have to think about if we want to understand the brain workings. And in one of the projects that we've worked on in the lab is we've taken a focus on local macrocircuits because of a collaboration with experimental work where they have been able to record these theta rhythms in an in vitro whole hippocampus preparation. So what that has allowed them to do is get some information about some cell types simultaneously with the ongoing population activity. So I see this as a giant puzzle where we have some pieces, and now we could use the mathematical model to bring it together and come up with ideas, hypotheses, predictions to explain how these rhythms will come about and such how they would be affected in disease. So I go back to the equations that I showed you before, because these are very famous equations, Hodgkin-Huxley. And I want to mention this was actually another Nobel Prize that was back in the 1960s. Basically what Hodgkin-Huxley and actually Katz also did was they took the squid axon, the giant squid, here you see a picture of, an axon or the nerve, the cell in this, that is a giant axon. And by a series of experiments, equivalent circuit representations, electrical engineering, and computers of the day, which was like a whole room, they came up with a set of equations that allowed them to describe how the firing of a cell, electrical firing of a cell, would occur based on the underlying biophysics, sodium channels, potassium channels, which are in these equations. I won't get into the detail, but suffice it to say is this set of equations that they derived and they did experiments to come up with the parameters, explain how the firing of a spike, an individual firing unit in the brain of the squid axon worked. And these equations are used, like we use them and many other people in computational today, to represent what's going on biophysically in an individual cell. So we have to figure out what they are for the different cell types and all the rest of it. So the point here is these are several mathematical equations. You cannot solve them with a paper and pencil. You have to write a program and a code to simulate what's going on. So this is immediately, if you're interested in neuroscience, there's many aspects associated with it, but you need to be able to do programming and coding to solve these set of equations to understand how the cells fire individually in a circuit and all the rest of it. So this is where the coding and the programming comes about in our work. And what I want to share is this image that one of my students made, because it kind of brings it all together in some sense. So we have this whole multi-scale, multi-temporal aspect going on in these complex brain organs. We have access to different pieces of experiments. We have an understanding with sets of equations that have been developed by many people and are being developed in different ways today. But you have the whole animal, whether it's a mouse, a human, or whatever. You have their brain. You have part of their brain, the hippocampus, and now we can build local circuits. So in our case, the CA1 circuits with cell types, and different experimental works allow us to know something about the cell types, something about how they're connected, something about the circuit. So in this schematic that my student Alex made, this is now focused on one of the cell types. But basically, in a nutshell, this comes all together in a computer. So many sets of mathematical equations that then we solve using codes and algorithms to come up with an explanation, to come up with an output. And because we have the model equations in hand, we can actually use it to understand what's going on and how the different cells produce theta rhythms in this case. And so really, it's always a balance. We have to build our mathematical models with experimental data, but we don't have every single detail because you can't do all those types of experiments, and definitely not in humans. So we have to make these translational ideas. So we build the models. In order to solve the models, we need to do computation. We need computer science, we need techniques, we need large computing, high performance computing to solve these sets of equations. But it's not like, you just solve it and you're done. You have to understand what's going on. So you need theoretical analyses to go along with this. So I have these arrows here because we need it all, and it's always a balance. And it's sort of like, what you're interested, where you do exactly how you fit in there. And so the mantra in our lab is to neither ignore the details nor to be consumed by them, because if we're trying to understand the brain, we're talking about networks, what the context and function is. Are we talking about sitting there paying attention, or maybe sleeping, as some of you guys might be, or you're walking around, you're forming memories? There's a certain context and function of what's going on at a particular time, so that has to be taken into consideration. So theta rhythms, as I mentioned, is important in navigation, spatial navigation, memory consolidation. Now, when you're doing the model, you have to think about the size of the model. How many cells? Hundreds, tens, hundreds, thousands? Of course, we have millions and billions of connections. How are they connected? What are those characteristics? So if we're going to make progress, this is the mantra. Don't ignore the details, but don't be consumed with them. You have to make decisions. And so that's why it's a balance. And one of the ways we've tackled this balance, just to share, is, as I mentioned, this theta rhythm and these experimental works that have gone in collaborators' lab, is we've built what I'm calling minimal models here. So essentially, there's sets of mathematical equations that represent, in this case, these are two different cell types that could generate this rhythm. Because it's a minimal model, we were able to use the model to explain how this rhythm comes about based on these two cell types. But it is minimal, so we're sort of stuck with all the different cell types and other complexities. So this gave us some sort of understanding. But what we've also done now is took advantage of a detailed model. So this model was developed in another lab, Ivan Chos's lab in Stanford, and this now has 9 different cell types, and they're connected in much more complex ways. So just to give you a sense, to solve the set of equations in this detailed model relative, this is order of 1,000 times different. So if you want to actually understand what's going on in this model, it's really challenging. You could understand it with this model. So what we did was we did a comparison of this model, of these two models, And then we took a slice of this model, this detailed model, that we could now explore with our computer programs fully. And now we have actually a biophysical explanation for the theta rhythm. So this is very much ongoing work, but what is very exciting about it is now we could think about cell types that change in particular ways in disease to potentially explain how the theta rhythm is changed. And I'm sort of in my last couple of slides, but just to give you an overview, so I've sort of tried to give you a sense of sort of the mathematical models, the computing and the program that's required to undertake this kind of work. But basically, the important thing is a cycle and it's a collaboration. So we're looking at modeling individual cells in terms of details, biophysics, and the structure. We're also looking at microcircuits. And I'm going to cycle the wrong way. It doesn't really matter. But basically, these are cycling, ongoing cycles. I mean, they never end because a mathematical model is always approximation to the real biology. So this has to go on. But the important thing is it feeds back. You don't just do a model and put it to the side. You want us to help us understand what's going on. And this, of course, requires collaboration because I don't personally do experimental work in my lab. And the idea is we want to converge and translate this understanding to the human system, because of course we cannot do these kind of detailed experiments in the humans. So we have to get an understanding and hopefully translate it to the humans. And we have done that to a certain extent so far. So the question is why, which I've kind of alluded to already. And so why? Just because I'm curious. I'm very excited about bringing math because I've always loved math since I was in primary school, high school, and the rest of it. I also wanted to be a vet, but I wanted to bring the math and biology together. So I feel very fortunate in terms of, if you like, my passion, which is sort of this combination that I've been able to sort of get to through neuroscience and computational neuroscience in particular. But of course, the big why is neurological disease. So we have many devastating neurological diseases that I'm sure you guys have heard about many. We heard about visual impairments in the earlier speaker, but we have Alzheimer's disease, we have Parkinson's, and we have many that are studied at the Krembil Research Institute. And what I wanted to point out here is so we know there's lots of, memory impairments, different things associated with disease, but just to figure out biomarkers, diagnostics, therapeutics, all the rest of it, we have to understand how rhythms that occur in the brain that are important for coding and memory consolidation, all the rest of it. What we also know is people have recorded these rhythms and shown that they change in very particular ways during disease. So Alzheimer's disease. So this is an individual cell in the wild type in a mouse that has Alzheimer's, and there's particular changes in this cell type. There's particular changes in the rhythm, which is a population activities. There's epileptic activity, change in cognitive function. Now at the whole brain level, you have deposits also and changes in what's called the default mode network. But the point is, by having these mathematical models and being able to solve them with computational techniques and lots of programming, we're able to hopefully bring our understanding together and help in sort of, these sort of devastating diseases. So that's the big why. But really, it's about what you are interested in doing. So with that, I'll stop and acknowledge, of course, funding in order for the work to go on. But all my lab members, because of course this is not me by myself doing this. This is some excellent students who have been in the lab in the past and at present, and of course collaborations and funding and support from our research institute, as well as lots of computing. So Neuroscience Gateway, SciNet, which is part of Compute Canada, heavily used to do a lot of the computing that's involved in our work. So with that, I'll wrap up and happy to answer any questions, which I understand will be at the panel discussion later. So thank you for your attention and your time.
Thank you so much, Dr. Skinner. That was really interesting. I loved how you kind of like summarized all this kind of components of, understanding a cellular level to a population level of how the brain works. And I really love that Photoshop of the brain on the Sky Dome or Rogers Center. That's how you can tell our ages, because we think of the Rogers Center instead of whatever it's called now.
Sky Dome. It wasn't Sky Dome then.
Yeah. But thank you. So thank you both our speakers. That was really amazing talk, and hopefully all the students have learned kind of how. So you saw from Dr. Reber how you can have a application based, so using kind of programming to develop therapeutics and applications to help people with visual deficits. And then you saw from Dr. Skinner, which is looking at more at a biological level, how do we use math and like what she said, coding and computer science to understand what is really going on there in healthy and diseased patients. So if our Science Rendezvous people can pull up the PowerPoint right now, that would be great. So now we have our career panel discussion. So if you guys have any questions at all, this is a great time to talk about, ask them about the careers, ask them about the science or the talks that you've had. And Eduardo and I will also kind of go through some of the questions that we saw on the Q&A, as well as our poll that we released pre our event. So yeah, I think in a real setting, I love panel discussions because you just sit at kind of like it feels like a fireside chat, with coffee and with wine. Maybe not for this audience for wine, but but maybe next year we can do all of this in person. So next slide, please. So as a icebreaker, because I kind of talked about islands and stuff like that before the talk, I just wanted to ask Dr. Reber, I guess you can go first, and then Dr. Skinner, if you had to, if you had to be in a deserted island, what would you bring and why?
That's an excellent question. I haven't thought about it actually. Maybe just paper and pen. I don't know. To write down what I experienced, to describe things that are around in my environment. Yeah, maybe I would bring this.
Yeah, that's a really scientist kind of way of looking at things. So great answer.
It's not very high tech for sure.
Yeah, I mean, most high tech things will require electricity and things like that. So, okay, Dr. Skinner.
Well, assuming that it's just one thing, I guess I would probably bring some sort of desalination, water desalination, because I'm in this island and I probably wouldn't last too long if I couldn't drink water. So I'm a very practical person, I guess.
Yeah, I'm also a nature person. Been looking into kind of like there's chemical pills. There's also like water filtration tubes, and that also goes into the whole innovation scheme that we're trying to bring to our students. Is that there's a lot of ways that we can kind of innovate and create products for different things. Okay, so Eduardo, we can start with maybe asking a bit of career questions, and then we'll go into some of your talks and some of the details that you mentioned. So Eduardo, you want to take it away?
Yep, for sure. So we'll start with some career questions that we've seen kind of going around. The students are very interested in trying to see hear more about your experience, how you got to where you are right now. Sort of like what were your experience in high school? What kind of courses did you take? And like at what point did you knew that this is what you wanted to do? I guess we can start with Dr. Reber to see what your experience on that.
Well, yeah, so I'm French actually, so up to high school, I am not sure that the system is exactly the same than in Canada, but I took science. I mean, it was like science classes, and I must say that it may sound stupid though, but when I was a kid, and I think you all know Commandant Cousteau, maybe, I don't know. Francis probably knows. A young generation, maybe a bit less. It was a like nature life documentaries and mostly sea life, and that was broadcasted on TV on Sundays afternoon. So it was like documentary about nature and sea life, and this guy had a huge boat and was basically cruising the world, exploring seas, and that was in the late 70s, 80s. I was a kid at that time, and when I was watching those documentary, that was just so inspiring. So I said, I want to do that. So I'm not a like sea person whatsoever. I'm not studying oceanography or things like this. But I think this is what, at one point, turned something on in my brain, saying like, I want to study nature. So that's how probably I found my path to biology. And then I studied at the French University in Paris, and I studied biology. And I'm trained as a molecular biologist originally. But this also shows you that now I'm doing neuroscience, and my background is more biology than computational or math. But I turned to mathematics a little bit later on. So this also shows you that during your career, you're learning all the time, and you may need to acquire new skills or new knowledge, even though you are 40, 50, or even older. And the type of job we're doing allows you to do this, so you can acquire new knowledge quick. It takes time, of course. I mean, and it requires work, but you can do it. And so you basically complement your knowledge, your skills throughout your life. And I think this is fascinating when you do science. And I think this is the major rule for every scientist. And I think Dr. Skinner will probably confirm this. So this is the beauty of the work we do. And then, yeah, so I did a PhD and then in France, and then I moved to the US, to the Salk Institute in San Diego. The first motivation wasn't really the science, it was surfing. So, because it's one of the best spots for surfing in the world. So I went there for surfing and also for science. The Salk Institute is probably one of the top 10 institutes in the world. And that's where I learned neuroscience. Originally, I wasn't a neuroscientist. And so that's where I stepped into the neuroscience field and discovered neuroscience. Again, it's a whole learning phase, and learning curve takes time. But if you get a chance to meet great people, inspiring mentors, then you can go find your own path there, and eventually find your own science and your own research projects.
Thank you so much. And so Dr. Skinner, if you would like to share your experience.
Sure. So yes, I agree with everything that Michael said, in the sense that, what's important is sort of your curiosity, your passion, your drive of what you find interesting, and don't pigeonhole yourself. We're always learning. We're students for life. I mean, that's what's so exciting and special, I think, about being able to be a scientist as your job, because you're always learning. We never know everything. And so it's a matter of reaching out. And you saw in one of my slides, I had those arrows. And so in my case, even though it's a little different from Michael, I started off, so actually, as I mentioned, I grew up in Trinidad and Tobago, and I came to Canada just to the end of, I'm actually Canadian by birth, but I came back just at the end of high school. And then my undergraduate degree was in applied math and computer science at Waterloo. So nothing to do with biology and math or anything like that. But I did actually want to be a veterinarian originally. And then because I loved math, we're starting a faculty of math at Waterloo, I sort of went for it. But then towards the end, I thought, well, what the heck am I going to do? I really like the biology. So I went actually into biomedical engineering, still not neuroscience yet. So it wasn't until my postdoctoral studies in the United States at Brandeis University that I got into neuroscience. And it's because I had fantastic mentors. This makes a huge difference, as Michael mentioned. I was in the lab of two very well-known neuroscientists, Eve Marden and Larry Abbott. And they were actually combining the mathematical modeling and the biological system. And so to me, I thought, oh, this is normal, but I realized soon after that it's not. And it's actually something that is, even though I was very excited, I sort of lucked out. And I lucked out there, we went there because my husband actually had a position at Harvard doing astrophysics. So it's sort of like, it's life. You try to go after what you're interested in, but you have your life. So you kind of go, you do stuff. And in my case, I was very fortunate to be in an environment that allowed me to get into neuroscience and really combine the math and the computer science and the programming and the biological system, actually do experiments and all the rest of it, and then carried on in California and all the rest of it before I came back. And now being at the Krembil and thinking of tackling disease through the mathematics is very exciting to me, because now we could think about our mathematical models, if applied and integrated in different ways, of really bringing about an understanding in ways that it would not be possible otherwise. So to students, so you're grade 5, 6, 7, you have your interests, ask questions, never stop asking questions, be curious, figure out where your passion is. And neuroscience is everywhere. So whether it's the math, the programming, the biology, it is what it is at the time. Don't pigeonhole yourself. Don't feel you can't change later on, but just be excited and interested and keep asking questions. I got in trouble for asking too many questions when I was in high school, but luckily I had a mom who supported me and told the teacher she should ask questions. So that's how you learn, and never be afraid to ask questions, because nobody knows everything. Okay, so that's what I would say.
Yeah, so thank you. I think like those both were really inspirational because it kind of showed how your career kind of transformed over time. And even for grad students or undergrad students, I think that's very reassuring because a lot of times we don't know where things are going to end up. And sometimes it's just follow our passion, I think. However cheesy that sounds, I think that's a great message for the students. So I wanted to ask a fun question. I guess I'm the fun question guy here. But so if you had to kind of choose a different kind of career field, what would it be? And I think maybe Dr. Reber, you might be a surfer or things like that, just from your story. But I think the reason why I wanted to ask this question is because right now, like with all these fields and all these science and innovation, everything is so interconnected, right? So if you can imagine yourself being interested in other things besides what you're doing right now, so you can still be a scientist but different fields, what would that be?
Am I supposed to ask us this question first?
Yes, Dr.
So still in the field of science, but something else. That's what you're saying.
Yeah, or if you can imagine a different career, what would it be?
I admire musicians. I'm myself playing a little bit of drums. I'm really bad at it, even though I've been playing for 20 years or even plus. But I'm just admiring those professional musicians, whether it's classic music and I would have loved to be one of them, a professional musician. I have one in my family, actually. I have one of my cousins is a pro drummer. So I'm admiring that guy because I think that would have been the second dream. I mean, the first one, well, I don't know if there is a one or a first or a second, but I had two dreams. Maybe one was to be a scientist, and I potentially partially achieved that. I would have loved to be a musician, yeah. And for the creativity aspect, but also for the technology aspect, I mean, recording instruments, nowadays you use a lot of electronics also to modify the sound, to reach like very pure sounds. When you have a beautiful instrument like famous violins, that are generating this pure sound, and you want to record that sound, and the recording makes it worse than it actually is. So you have to work on the recording technology to make it as pure as it comes out of the instruments. So all these, yeah, this would have been a nice life maybe in the future.
I think it's also fascinating because your work where you showed you're trying to develop this neuroplasticity, you're trying to create networks in your brain that is pairing kind of the visual with the auditory. So it has a lot of connections with this too. And I think, so back at McMaster, I studied psychology before. I'm an immunologist now, which is really random. But they had a lot of kind of music, cognition, music psychology, music neuroscience, whether it's in therapy or trying to understand how the brain perceives things and how it networks. I remember this one time where they showed a person with Parkinson's disease, and then you turn on certain specific types of music, and because of the rhythmics and the beats, that can completely change the motor system and how it functions. And so I think that was really exciting. So it's cool to see these connections, and that's why I'm kind of asking these questions to see, if we're, as scientists, we're not just scientists, right? We have other hobbies and interests.
We are real people.
So Francis, sorry, Dr. Skinner, do you have what career would you be?
So I had mentioned about wanting, when I was in high school, wanting to be a veterinarian. I loved animals and stuff, but I think if you to answer that question today, I wouldn't probably say veterinarian. I think the brain and the mathematical, these things are very exciting to me because we're all these individual people, and we're a bunch of memories, and sort of trying to sort of think of that at sort of, I'm not studying it, but at a philosophical level. And you know, all the stuff we see in the world today, like people, everybody is an individual. Doesn't matter what country you're from, what background you're from, where you grew up. We're sort of, you know, should be sort of recognized as unique individuals. And so I think, you know, as we learn more about the brain and the human body, and we sort of see, you know, in the move towards personalized medicine, we see these kind of aspects there. And so I think if I had another career, I really have a huge admiration for people who are, say, in social sciences, because, you know, understanding and trying to sort of help people in those all these sort of individualized situation, it takes amazing strength and resilience. And I don't think I could do that, but I really admire people who do that. And perhaps that's something I could do a little bit of more of, you know, in my spare time. But yeah, I see it, you know, because it's not just science. Science is not, it's science, humanities, we're all together in this, right? I mean, you know, if there's sort of more interaction and understanding, then I think, you know, we see everybody's strengths as individuals, no matter where they are. So yeah, that's my short, maybe not sort of fun answer, but maybe getting a little bit too serious, but yeah, that's what I think.
No, that's a great answer too. And I think that also goes back to your presentation about understanding neuronal activity and going into a bigger kind of regional dynamics of the brain. And now you're looking across different brains, you know, how do people connect and how do everyone come together? And also the application side, I think, is fascinating too, because with behavior, we're just so complex as beings, but how is all that kind of determined just by like cellular one-to-one signaling? I think it's fascinating.
Mm-hmm.
Yeah.
Yep.
Eduardo?
Yep. So going back to what you, I guess what you both mentioned about science just being a more interdisciplinary type of experience, and just basing off on your backgrounds, how you've gone from different fields in science and putting everything together. I think one thing that we would love to know is more regarding the computer science aspect, the programming aspect, how it's been integrated in your own research, and kind of maybe like a take on how computer science or programming in general is growing or is developing in the field of science, or even in Dr. Reber's case, in the clinical field. So Dr. Reber, if you have anything to say about that.
Yes, sure. I mean, as you can see, what I presented is part of the work. I mean, we also do some mathematical modeling, not to the extent of Dr. Skinner's work, but definitely computing, programming is everywhere in science. And I think nowadays we couldn't even work without any sort of computing. We have big data analysis, we have deep learning, we have artificial intelligence. All this relies on heavy computing, right? So I mean, whatever you do or you study computer science, doing biology, for example, or medical research has a very strong component of computational work and computational aspects, definitely. I mean, what we do in the lab, I'm some sort of coordinating and making the link between what we know from the biology and how we can use codes, computational work, VR, and 3D Unity to somehow stimulate your vision in ways that are biologically relevant. And so I'm not coding. I'm not the guy who writes the codes. I mean, Eduardo is, and Pi is. But basically, I tell them what I'm expecting in terms of stimulation, and they transform it so that they can encode it and write codes and generate the actual visual task or visual stimulation program. So Dr. Skinner would probably be more appropriate to answer that kind of questions as a computational and scientist and mathematician. But yeah, I mean, I truly believe that we are not going to step back from that. I mean, computing is going to be more and more represented in science, in biology, science, but I guess it's the same in physics, in chemistry, and all other sciences, and social science too, you know, because they are collecting tons of data nowadays, and we need powerful calculation to compute and analyze those data.
Yeah, I totally agree. And as being part of that experience, I really think that it's really the future in terms of medicine and science itself. And it's going to be something that's going to be required. But Dr. Skinner, I think you having such a big experience in computer science, I think you could probably adjust to that as well. Attest.
Yeah. So yep. So I mean, I think similar and different to Michael. So similar in that I myself, I'm not probably doing the hands. I'm doing very limited hands-on computing nowadays. It's the students in the lab, but certainly when I started, I was doing all the coding and the mathematical equations, and I still sort of course, oversee it and in that extent. And what I would say is, whether you're in computer science or not, I mean, there's many ways to kind of learn coding and programming nowadays. There's so much online, the path, and you're self-teaching, and you should definitely make the effort because it is in everything. And understanding sort of the fundamentals, I just push a button and something magically happens. Sort of getting some sense of what's under the hood can only serve you well. So whether you want to sort of be a hardcore programmer and coder, doing, actually developing the sort of packages and the platforms that other people use, or you want to be just a user. But I would really hope that people don't be blind users, because that's dangerous. So definitely, if you love it and spend lots of time, or you only partially like it and you want to use it in other ways, do coding and programming and understand what's there. I mean, my daughter, who's gone through a biological program, she's now doing sort of learning about programming, because she realized to analyze all her images of cells, she needs to kind of do some coding. So whether you're in the biological side or you're starting the computer, AI, whatever, it's worth the effort to sort of, do some kind of develop some in-depth knowledge of programming and computing. So, I happen to have a little bit more in-depth knowledge because, my undergraduate degree was in it. But nowadays, I mean, we learned Fortran and other things like that. And then I self-taught myself many other languages. I'm certainly not an expert in Python, but, hopefully I'll learn that a little bit better when I get a chance. But the point is to understand fundamentally what it is about. Don't just push the button and think magic happens. Really learn about coding no matter what field you're in, whether it's a more biological field or more heavy duty, AI sort of industrial side, or then you'll probably be doing it directly. But yeah, so maybe I'll leave it there. And if people have more specific questions, yeah, but it's.
Yeah, and that's what we're trying to kind of accomplish here in this competition as well, is to kind of introduce these skills to these students so they can later on apply them to whatever career they want to undertake. But like you mentioned, we do want to move on to more specific questions. So now we move into the Q&A part, where we're going to answer some of the more specific questions that the students have for either one of you.
Yeah, so I guess I can start with Dr. Reber. Sorry, you're always our first go-to person. But regarding your talk, again, I thought it was really, really cool that you developed an app base where you're trying to connect kind of, or coach, like what you said, to develop the neurons in areas that are deficit. There are quite a few questions about, is it possible to, so you mentioned all these different kind of diseases, some are the brain, some are in the eyes, and some are even in the optical nerves. So is there procedures where people can alter or transplant optical nerves? Are there ways to have a projection? I guess this is more futuristic, but have manipulate the electrical signal so you can project images to the brain directly. I remember there was a paper way back then talking in mice where they were stimulating the hippocampus to recreate memories that did not exist in mouse. So things like that, I think, are starting to become like a reality. So any comments about that?
Well, yeah, I mean, so far, transplanting things in the brain is not really possible. I mean, we cannot transplant optic nerves. The reason is, well, you would have to connect the wires the right way at both ends. So if you transplant an optic nerve, you would have to properly connect the wires to the eye and also to the brain. And those wires are not randomly set up. I mean, there is a very defined and very precise organization of the wiring of the visual system. The picture that I showed, where you see this diagram with all the wires in the brain areas, this is something which is established during development, and it's very precise. And nowadays, we can't recreate this so far because we don't yet understand all of the mechanisms there. So when I said at the beginning of my talk that one of our research projects is about helping people with low vision, it is because we have other research projects which are much more geared towards these basic aspects, these basic processes that are happening. And so that's why Dr. Skinner mentioned that the brain is the most complex organ of our bodies. So I mean, we are still far away from understanding how the brain works. We are at the beginning, you know, we are at the baby steps, I would say, of understanding how the brain works. It's extremely complex, it's extremely complicated. And no, we cannot replace eyeballs, we cannot replace optic nerves for now. However, there have been studies that show that you can transplant in the retina some sort of CCD cameras that would activate or would stimulate some of the cells in the retina. And a recent paper actually came out related to a blind person that is now able, through that type of mechanisms, which is very heavy and very complicated, because basically you are wearing glasses. In the center of those glasses is a camera that is basically filming your entire environment. And then you have a cable that is going into your eye, some sort of, and stimulates that tiny little chip that is lying in the back of your eye, which by electrical stimulation is then stimulating your optic nerve and your visual brain areas. But the max we are now able to reach is basically able to discern shapes. So for example, on the slide that is shown here, you know, what is your story? A patient with such device would be able to basically see the frame of the window here, and maybe the frame of the tiny little windows on top of that. That's basically it. So it's very, very limited so far, and we are far from having something very reliable and that would correspond to the vision we actually have naturally. If this answered your question.
Yeah, I think it did. And that was really fascinating. I didn't even know that those kind of technologies are being kind of tested on. I guess the next question is, so when you were talking about just right now about all the different kind of networks and systems that are connected into the visual cortex, even in areas that we didn't normally kind of knew. So with your application that you're working with, Eduardo, where you're trying to coach with neuroplasticity, like, do you have plans on doing EEG or other kind of electrode kind of signaling to see? I don't really know how you can actually track plasticity over time, but kind of transplanting that visual behavioral thing you're trying to achieve therapeutically with the biology.
Well, yes, I mean, that's as I said, I mean, for us, it's really the beginning of a story, which I hope will be a long and exciting story. The idea would indeed be to record what the patient actually, or I mean, to record the brain activity of those particular patients using maybe EEG, but also coupled to another technology, which is like fairly, or it's not new, but it's very convenient to use at home like this for those patients, which is called near infrared spectroscopy. So it's basically like the EEG. You are recording electrical waves in your brain, and this connects to Dr. Skinner's work. You're basically recording brain activity, and there are different ways of doing it. And the near infrared is a technology that uses light to do this. It's very convenient to use because it allows you, or it tolerates movement and so on and so on and so on. Whether EEG, when you do EEG recordings, you have to really stay still and not move your eyes and so on and so on and so on, which would be difficult then eventually to interpret the data when you use it in a visual stimulation task. So in an ideal world, and I hope we will get there as soon as we get the financial support, I would say, is to combine visual stimulation with the VR headsets together with this brain recording technique. Now we are also waiting, I would say, for an upgrade of the Oculus Quest, and we hope soon they will install an eye tracker into the Oculus, so a built-in eye tracker into the Oculus Quest. And from what I've heard, and Eduardo also knows that better than I do, but maybe in the coming month, or maybe I hope in a year or so, Facebook might come up with an Oculus Quest 3 with an eye tracker, and this would be extremely helpful for us because it will provide a lot of information about how the patients, how the people basically use their eyes to track those elements. You know, when we ask them to, for example, I told you about this fixation point that we have, this green fixation point, whether they are able to control their gaze, their fixation, while they'll track the target. So this will provide us valuable data and measures about the strategy used by the patient to track the target.
Yeah, awesome. So one final question before we kind of move on to Dr. Skinner. There was a question here, which I'm not sure if it was from grades 5 or 6 or sorry, 6, 7, or 8. But the question about funding and the question about, you know, where do you get research funding in general as a scientist or for this project, but also like what you said with how your work is, you know, really taking technologies that are developed by companies like Facebook and stuff. So how does that work with your integration as a researcher with these startup business / big companies / tech industry, and also where the funding is going?
So the fundings are from different sources. I mean, there are the official governmental fundings like CHR, like NSERC, and many other funding agencies, whether they are in Canada or US. And then you have donations, you have donors, and at Crumble we have there's a strong activity of donations there, and there is a whole group and department of people trying to raise money for to support our research, but also clinical research and the work done in vision. And so basically, when you have so very innovative and unique research project like this one, you can only start this type of project with, I would say, a donor money. You know, because if you would apply for a grant at CHR or at NSERC, you need to have so-called preliminary data. So if you go through official agencies, you must show them that even though your project is risky, you already have some preliminary data that tells us that it's doable, it's feasible. You know, you're not going to start something totally crazy, which after 2 months collapses because it's totally irrelevant or it doesn't make sense at all. So you every time need preliminary data when you want to submit grants and to obtain money to run your research projects. But to generate those preliminary data, there are a few ways to get this money. I mean, it's not a lot of money that you need to generate those preliminary data, but you still need to get it. And the easy way for us at Crumble is the donations and donors that show support for our research, for the doctors working there. And then using that money, we can I would say, jumpstart those projects and acquire enough preliminary data to then move on something bigger. Now, I must say that Facebook and big companies like this, we try to connect with them and to tell them whether, you know, on their side they would help by providing, for example, those devices or for free, for example, or basically helping us or listening to our needs that we may need to develop. I mean, within the device. But I must say, so far we have been rather unsuccessful in attracting their attention. I think. We are not money makers, so big companies are not very interested in our work, although we are using their devices to maybe on the long term, maybe provide some sort of support to patients, So yeah, money is definitely key. You have to think different ways of jumpstarting those projects and try to get the money. But we have very strong support from our research institutes and from UHN, and they love when we come up with such very innovative and risky project, and they support this.
Yeah, awesome. I think like in the future, it'll be amazing if somehow the tech industry and scientists and clinical doctors and everyone can just sit down and work together. It's more of a fantasy, but it would be really cool.
It would be
okay.
ideal, yes, but unfortunately for now, that's not the case.
For sure. And then Eduardo, so we have a couple of questions for Dr. Skinner as well, and then we're going to end our innovations speaking series #1 around 12:30. So.
Yeah, so Dr. Skinner, going back to your work about creating these models to understand how these neuronal networks work and how the input affects the output, a lot of the students seem to be curious about, or some students seem to be curious about, like the rhythms that you mentioned, about how these rhythms change, or what makes these rhythms change. Are they the same? Are these rhythms like fired every time a neuron is activated? Is it possible you can talk a little bit about that?
Yes, sure. Yeah, that whoever asked those questions, you hit the nail on the head. So this is hugely challenging, because when we talk about rhythms that I showed you sort of in some of the earlier slides, these are rhythms that are recorded by not just one cell, but many cells together in a network. And so in order to actually see it on your recording electrode, several individual cells sort of have to come together in a synchronized or coherent fashion to create this rhythmic activity. And this we know occurs at different frequencies, and the theta frequency that I was referring to particularly, which is about 3 to 12 hertz, so about 100 millisecond time period, is shown to be important, maybe underlying plasticity and all the rest of it. But there's other rhythms that are higher frequency. So there's, for example, a ripple frequency that's 100 to 100 and maybe 200 hertz. There's slower frequencies, delta rhythms that you see during sleep. So they're different frequencies, and it's not as if you're changing the cells in your brain. So all these different cells behave differently under different situations, different contexts, to produce these rhythms of different frequencies. And so as I sort of explained to a certain extent, the theta rhythm, it is important it seems to be part of the brain code based on many studies that other people have done. And these higher frequencies, fast ripples as they call, seem to predict seizures in the brain, for example. So there's a lot of interesting, many studies that show that rhythms of particular frequencies are important. So really, how do these different frequencies actually come about? Well, the short answer is we actually don't know. There's many ideas and speculations and models, of which we do some of them, produce these rhythms. But just because you produce it in a mathematical model doesn't mean that's how the brain actually produces it. And so really, that's why these sort of cycles that I talked about is, these two directional arrows are really key. So you come up with ideas about how these different cell types contribute to the theta rhythm, for example, as we're doing. And now, because of advances in molecular biology techniques, genetics, you can actually target specific cell types to see if the predictions and suggestions and hypotheses of how they control these rhythms from the whole network, of which that cell is one type, is correct. So this is what I mean about the cycling and the testing. And because we know certain cell types, so for example, there's a cell type called the Chandelier cell in the brain that seems to be particularly infected in schizophrenia, but it's part of the whole circuit and it changes certain rhythms. So I'm trying to give you a sense of these frequencies and these rhythms. It's a huge question. And so I don't have the answer, but I could say that this is one of the things we're trying to understand by incorporating cellular aspects, because I think we have to. Other people are or not or doing different things, and it's not what's right or wrong, but it's in terms of bringing them together and cycling back with the biological system. So this is where sort of a lot of theoretical insights are very helpful, in that how you get this coherent synchronized activities from individual cells that have certain, if I think of it of a mathematical model, if I have a mathematical model that represents a cell And then I could use, write a program that solves those set of equations. I change the parameters. It changes how that, excuse me, how that cell affects the frequency that comes out. It could increase or decrease the frequency. And now we can actually go in and do stimulation. So Dr. Reba was talking about different stimulations that can be used. So it matters what frequency of stimulation you use. I mean, these are people are doing these kind of studies to show you could enhance, say memory or whatever by a certain frequency of a rhythm. I mean, these are all sort of test things that are going on. But does it make a difference if you target certain cell types, certain brain structures? I mean, deep brain stimulation, Parkinson's, people try to sort of figure out which brain structures, which regions, which parts matter. So it's a huge question. It's a very exciting field. The rhythms are at the population level when we record it, and it's how the individual cells with their properties, individual structures, individual biophysical characteristics come together to produce this output of certain frequencies. So like I said, there's many ideas and speculations out there coming from a lot of these mathematical modeling and computational techniques, but we don't know. The only thing we know is it matters. So, and it seems to be important. So that's why it's about the youth, So if you find this kind of stuff interesting, really, we need all the help we can get is how I see it. And so, like I said, I've been very fortunate students in my lab so far, but there's a lot, as I said, and as Michael said, I mean, it's a very complex organ, and however you study it, the rhythms, whether you look at it from actually developing the technology to apply certain frequencies, I mean, this is also going on in different levels, but you need to understand what's there, because why does a high frequency stimulation help DBS, help Parkinson's disease patients, for example, in certain structures, but not slower rhythms? But if we apply a different region, maybe a certain frequency is more and target certain cell types, maybe that's going to be more efficacious or more helpful for people with Alzheimer's. Actually, we know where it starts in the hippocampus. So hopefully that sort of answers the question to a certain extent.
Yeah, and I think that kind of follows up to the next question, because indeed it is a very complex thing to kind of model or try to represent even through computational means. And a lot of students were kind of wondering about like the setup, kind of like what type of computer setup are used in order to handle all of these calculations, and also maybe like kind of programming languages are kind of popular in order to achieve, or what data scientists are using right now to create these models.
Yeah, so I would say it's what works, in the sense that so as I mentioned, when I did computer science, I was learning Fortran and Pascal, different techniques, which is not really used today. Now Python is very popular. There's also like more sort of Java platforms that you kind of don't really need to write the program so much yourself. You sort of use it. So I think the answer is more what aspects you want to be involved with, and then you sort of learn what you need to learn in order to sort of solve the equation. So I think what is important to understand is how computer programs actually work. So, you write your code in whatever language, whether it's Python, Fortran, C, C++, whatever. They all have a similar structure, pseudocode, if then loops, while, understand sort of how it works in a fundamental sense. So it doesn't really matter exactly what language I would say, but once you have an understanding of computer programming structure, pseudocode, then it's what platform you use. It's sort of, that's not the most critical thing. It's more to understand you have to compile a program. You write a code, you compile it, what goes on in the background, and then you execute the code. So just if you push the button, just kind of know what's going on when you push the button is sort of really what I'm sort of advocating. But the program itself, what it does could be many things, right? It could be solving a set of equations like the example I showed you. So the language, whether it's C, Python, MATLAB, it sort of is not critical, because if you have that understanding, you can learn it. Anybody. I mean, as Michael said, right? He's coordinating, but I mean, I'm sure he can write programs, if he had the time to focus and do that, right? So, I'm just trying to teach myself Python these days, but I mean, that wasn't what I learned from, but I can understand code and go through it because I understand sort of the structure of code. So that's what I would say one should do. Not so much worry about, I should learn Python, I should learn, understand sort of the essence of what a computer program is and how it works, and do that through whatever language you want. I mean, I think Python is probably a good one these days. But yeah, there's many people are developing a lot of platforms, so you don't have to worry about it so much. Like the language, you just sort of take a mathematical model and put it into one of these platforms, and then it solves it and spits out the answer. So there's a lot of that being developed in computational neuroscience. But even if you're just pushing the button, please try to put the effort to understand about the program itself, because then it's a black box and it becomes a little dangerous. Okay, I'll leave that there.
Yeah.
Is that helpful?
Awesome. So that actually gets us to a good segue. So next slide, please. So thank you everyone for attending, and especially Dr. Reber and Dr. Skinner for those amazing presentations. I definitely learned a lot. And like what they've been kind of encouraging is, if students, whatever grade you are interested in learning coding, we do have a workshop held by our own Eduardo and our co-chair Sarath, who will be doing based off of Microsoft MakeCode, which uses a Scratch-based kind of programming, so very basic. But you'll learn those kind of different structures and things like that. So come join our events. We also have a bunch of other things going on. So go into our website, sign up today. And yeah, so next slide. I would like to thank everyone that's involved. So especially our organizing team for the hackathon. It was a huge, tremendous, like it started from a whiteboard idea, and now we've gained 250 participants plus other public members as well. And we've have amazing speakers and amazing guests that are incoming as well. And so thank you, Mimi, Eduardo, Adiz, and Blake for all the work that they've been doing, and also the rest of the Science Rendezvous team, especially the promotions team, our chairs, Sarath and Trin, and Lily and Amy for other things as well. Next slide. We also have a I Am a Scientist kind of program, and this is basically featuring a graduate student and a researcher in different fields, explaining about their lives, kind of like what we did here with Dr. Reber and Skinner, except on a YouTube kind of platform. So check us out there. Next. Thank you all our sponsors, including Kremble and NSERC. Without you guys, this stuff wouldn't be possible, because even though it's virtual, there are a lot of costs associated with it. Thank you for University of Toronto, as well as our Science Rendezvous larger kind of branch, as well as our individual U of T team. Next. And that is it. So for students, I know it's a long day, so please go grab lunch, go grab food. We're going to start our opening ceremony. This is really, really essential for all the details about the hackathon competition. So please join us there. For other people, our next workshops are June 12th to 13th. And this specific Innovation Speaking Series 1 will be posted onto our YouTube for everyone else to see. And so thank you again for our amazing guests, our Science Rendezvous helpers today, including Eduardo, Blake, Sophia, and Emily. And we'll see everyone soon. So thank you again.
Thank you all. Bye-bye.
Thank you.